studying the structure of a prokaryotic cell, we know that in prokaryotic cell there are no membrane-bound organelles. The organelles in the cell are not bound by membrane. So the first structure in the prokaryotic cell is the cell wall. As we can see, the cell envelope is the outermost covering of the cell. So the first is the cell envelope. Now this cell envelope is divided into 3 layers. It has 3 layers. The first being the outermost one is the glycocalyx. So let us talk about the glycocalyx. Glycocalyx, as we can see, is the outermost covering of the protoplasm. Whatever lies internal to the cell envelope is the protoplasm. So glycocalyx is the outermost covering of the protoplasm. The glycocalyx in turn is made up of 2 layers, an inner S layer and the outer layer of mucilage. So we have glycocalyx, which is the outermost covering of protoplasm. And it is divided into an inner S layer. Now this inner S layer is made up of polypeptides which are rich in amino acids. So the composition of the inner layer is polypeptides made up of amino acids, rich in amino acids, and the outer layer is the outer mucilage layer. And this outer mucilage layer is rich in carbohydrates. Now when this outer mucilage layer is thick, it forms what is called as capsule. So when thick, it forms a capsule. Or it is called as capsule. Now what are the functions of this outermost layer of glycocalyx? Glycocalyx functions as, the first function of glycocalyx is that it prevents drying of the cell. That means it prevents desiccation. Second function is that it being the outermost layer or outermost covering, it protects. So it provides protection. So it protects from viruses, protects the cell from viruses, also protects it from phagocytes, protects it from chemicals. Third function is that it serves as a site of attachment. So it provides attachment. And the last function of the glycocalyx layer is that it provides virulence to the bacterial cell. So it is the layer which is responsible for virulence. So this was the outermost layer in the cell envelope. We now go on to the second layer of the cell envelope, which is the cell wall. So we have at B layer called as cell wall. We can see in the diagram that cell wall lies inner to the glycocalyx. At the same time, it is the layer which lies between the glycocalyx and the innermost layer of the cell envelope, which is the plasma membrane. So I can say that it is inner to glycocalyx and it lies between glycocalyx and the innermost layer of the cell envelope, which is the plasma membrane. Now inner to the cell wall is the plasma membrane, and there is a space between the cell wall and the plasma membrane. That space is called as periplasmic space. Now coming to the composition of the cell wall, what is it composed of? The cell wall is mainly composed of peptidoglycans. So we have the peptidoglycans. Along with peptidoglycans, it also has lipids. It has amino acids and it has fatty acids. Now bacteria like Mycobacterium and Nocardia, they have long chain fatty acids. These long chain fatty acids are called as mycolic acids. So we have Mycobacterium and Nocardia, which show long chain fatty acids, and these long chain fatty acids are called as mycolic acids. Now, what is the importance of this layer of cell wall? Now, it is the cell wall which reacts to what is called as Gram stain, and based on that reaction, the bacteria is called either a Gram positive bacteria or a Gram negative bacteria. So we have the cell wall which is responsible for reacting to Gram staining. And on that basis, the bacteria may be either Gram positive or a Gram negative bacteria. Now going on to the third and the innermost layer of the cell envelope, which is the plasma membrane. So we have at C the cell membrane or the plasma membrane. Now this plasma membrane is the innermost layer of the envelope. It is made up of proteins and lipids. Two layers of lipids and proteins form the plasma membrane. So I can say that it is made up of a bilayer of lipoproteins. Coming to the functions of plasma membrane, the plasma membrane is a very important structure. And the functions, first function is that it serves as a barrier. Secondly, it is the cell membrane or the plasma membrane which is separating the bacteria or which is separating the bacterial cell from its internal environment and the external environment. That means it is separating the internal environment of the cell from its external environment. So it separates the cell or separates internal environment of cell from the external environment. Now it is the plasma membrane through which two cells communicate with each other. So I can say that it is responsible for communication between two cells. When the communication is occurring between two cells, it is called as intercellular communication. So it is responsible for intercellular communication. Now it is the plasma membrane which has receptor sites to receive molecules and to react to those molecules of chemicals. So it has receptor molecules to receive chemicals and to react to them. Now these chemicals could be anything, even things like medicines. So these are the functions of the plasma membrane. Let us now go back to what we have studied about the cell envelope. Cell envelope is basically made up of three layers: the glycocalyx, cell wall, and the cell membrane. About the glycocalyx, it is the outermost covering of the protoplasm. The glycocalyx is in turn made up of two layers. The inner one is called as the S layer, which is made up of polypeptides which are rich in amino acids, and the outer layer is of the mucilage, which is made up of carbohydrates. When this outer layer of mucilage is thick, it forms a covering called as the capsule. The functions of the glycocalyx is that it prevents desiccation or drying up of the cell. It protects the cell from viruses, from phagocytes, and from chemicals. Also, it provides site of attachment, and it provides virulence to the cell. Coming to the cell wall, which lies inner to the glycocalyx, and it lies between the glycocalyx and the plasma membrane. The space between the cell wall and the plasma membrane is called as the periplasmic space. The cell wall is made up of peptidoglycans and lipids, amino acids, and fatty acids. In bacteria like Mycobacterium and Nocardia, they have long-chain fatty acids which are called as mycolic acids. These mycolic acids are a component of the cell wall. Now, it is the cell wall which reacts to gram stain and indicates whether it's a gram-positive or a gram-negative bacteria. Next is the cell membrane, the innermost layer of the cell envelope, which is called as the cell membrane. It is a bilayer of lipoproteins. That means it consists of two layers of lipids and proteins. The function of the cell membrane is that it serves as a barrier. It separates the internal environment of the cell from its external environment. It is responsible for communication between two cells, and it has receptor molecules to receive chemicals, and they can react to it and react to those received chemicals. Also, it acts as a site for metabolic processes. In the next session, we shall continue with the structure of the prokaryotic cell. That is, next is the cytoplasm. We have been discussing the structure of a prokaryotic cell. We have already seen about the cell wall. Now let us go to the next structure of the prokaryotic cell, which is the cytoplasm. Now the cytoplasm in a prokaryotic cell and also in a bacterial cell is granular. It is granular because it has many ribosomes present in it. Also, in cytoplasm, there are no membrane-bound organelles. There are only some structures which are membrane-bound, but the proper organelles do not have a membrane outer to them. The cytoplasm here in a prokaryotic cell again does not have sap vacuoles. The vacuoles which are present do not contain sap. There are gas vacuoles instead. Here, the cytoplasm is semi-liquid type or semi-fluid. So I can say that the cytoplasm is granular because of ribosomes and it is of semi-fluid type. It is semi-fluid, but it does not show any streaming movement. So no cytoplasmic streaming is seen. And there are no membrane-bound organelles. So cytoplasm contains organic and inorganic material or substances. Also, there are no sap vacuoles. Instead, what is present are the gas vacuoles. Now, as I have mentioned, cytoplasm will show certain structures which are membranous. There are two such structures. So let us talk about them. Membranous structures seen are, the first is called as mesosomes. Now, mesosomes are basically invaginations or infoldings of the plasma membrane. So these are formed from the plasma membrane. There are two types of mesosomes, septal and lateral. The septal type of mesosomes attach the nucleoid, which is the unorganized nuclear material, to the plasma membrane. So the septal are a connection between the nucleoid and the plasma membrane. This was the first type of the mesosome. Second type of mesosome are the lateral types. In the lateral type, the lateral type of the mesosome is not connected to the nucleoid. Now, what are the functions of the nucleoid? What are the functions of the mesosomes? The first function of the mesosome is that it is involved in formation of the cell wall. Very important function. Involved in formation of cell wall. It is also involved in replication of DNA. It also takes part in respiration. So these are the functions of mesosomes. Going on to the second membranous structure, which is the chromatophore. Now, as the name suggests, chromato means color. So that means it is these structures which have pigments. Pigments are the material which gives color. They are the substances which give color. So these are pigment containing. These are internal membrane systems. So what type of pigments would they have? They will have photosynthetic pigments. So when they are having photosynthetic pigments, they will be seen only in bacteria which are capable of carrying out photosynthesis. So chromatophores are present in photosynthetic bacteria and cyanobacteria. So they are seen in cyanobacteria and photosynthetic bacteria. So what are the types of pigments present in the chromatophores? There are pigments which are called as bacteriochlorophylls. There is a pigment which is called as carotenoid. And another pigment that the chromatophores have is called as bacteriophyocytine. So this was about the cytoplasm in a prokaryotic cell. Coming to the next structure, which are the ribosomes. So we come to structure number 3, that is ribosomes. As mentioned earlier, the cytoplasm is granular in the prokaryotic cell because of presence of ribosomes. So what are basically ribosomes? Ribosomes are made up of ribonucleic acid and proteins. So it is the RNA and proteins which are the main component of ribosomes. Now the ribosomes present in the prokaryotic cell are of the 70S type. When I say 70S type, here S stands for Swedberg number, or it is the sedimentation coefficient. Further, now this 70S type is again made up of 2 units. The 70S type is made up of a larger unit, which is 50S, and a smaller unit, that is 30S. So the 51 is larger and the 30S is smaller. Further, the ribosomes present in the cytoplasm are of 2 types. They may be either fixed or they may be free. So we have free ribosomes and fixed ribosomes. Now the free ribosomes are lying in the cytoplasm. So these help in protein synthesis. Now protein synthesis is the main function of ribosomes, but the free ribosomes help in manufacture or synthesis of proteins, which are then used for intracellular functions. And there are another type which are called as fixed ribosomes. Now the fixed ribosomes help in protein synthesis, which are then transported to outside the cell. So we have these 2 types of ribosomes. Main function, as I mentioned earlier, is protein synthesis. So that was about ribosomes. Going on to the next function of, or next structure in the prokaryotic cell, which is the nucleoid. As you can see in the diagram, the genetic material in the prokaryotic cell does not have a covering outer to it. The genetic material lies naked because there is no outer covering to it, and secondly, the genetic material is not associated with histone proteins. So the genetic material or the nucleus which is unorganized in the prokaryotic cell is called as a nucleoid. It is also called as the small chromosome. Now, genetic material here is present in the form of a double-stranded circular DNA. So we have a single double-stranded, single circular double-stranded DNA. Now this strand of DNA, or this single double-stranded DNA, is looped and coiled by proteins and RNA. So RNA and proteins coil this DNA. Now the type of proteins associated with the DNA here are the polyamines. There are no histones present in a prokaryotic cell. So polyamines are the type of proteins. And because there is no histone present here, the DNA is said to be, or DNA is called as being naked. So the DNA is naked. So that was about the fourth structure, that is the nucleoid in the prokaryotic cell. Let us revise through and go through the diagram also at the same time. The second structure of the prokaryotic cell is the cytoplasm. The cytoplasm in the prokaryotes is granular because of presence of ribosomes. It is semi-fluid. The cytoplasm is also called as the ground substance or the matrix. There is no streaming movement of the cytoplasm seen, and there are no membrane-bound organelles. There will be certain membranous structures. Organic and inorganic substances are present in the cytoplasm. Sap vacuoles are absent, only gas vacuoles are present. Some membranous structures that we see in the cytoplasm are mesosomes. Mesosomes are infoldings or invaginations of the plasma membrane. And there are two types of mesosomes, septal and lateral. The septal ones connect the nucleoid and the plasma membrane. The lateral are not connected to the nucleoid. Functions of the mesosomes is that they are involved in formation of cell wall, they help in DNA replication, and they also help in respiration. Chromatophores are the second membranous structures found in the cytoplasm. These are pigment containing structures, mainly photosynthetic pigment. So chromatophores are found in photosynthetic bacteria and in cyanobacteria. The pigments present here are mainly bacteriochlorophyll, bacteriophyocyatin, and carotenoids. Third structure in the prokaryotic cell is the ribosome. The ribosomes are made up of RNA and proteins. Mainly 70S type of ribosomes are present. Main function of the ribosome is protein synthesis. The 70S is made up of two units. The larger one is called as 50S and the smaller one is called as the 30S. Here S stands for Swedberg number or sedimentation coefficient. Ribosomes may be of two types. They may be fixed or the free type. The next structure is the nucleoid. Nucleoid is the genetic material which is in the form of a single circular double-stranded DNA, which is again long. It has a good length. It is coiled by or looped around by RNA and proteins. The type of proteins associated here with the DNA is the polyamines. The DNA is not associated with histones, and the DNA does not have an outer covering to it. So that is why the DNA here is called as naked DNA. Further, we shall go on to the next structure in the prokaryotic cell. Discussing about the structure of the prokaryotic cell, we go to the next part of the prokaryotic cell, which are the inclusion bodies. Now, inclusion bodies are certain structures in the cytoplasm which store some granules. So these are basically storage granules, and they lie freely in the cytoplasm. These inclusion bodies are of three types. They may be gas vacuoles, they may be inorganic or organic bodies. So we go to the first type, which is the gas vacuoles. Let us see what are gas vacuoles. As I already mentioned earlier, there are no sap vacuoles present in the prokaryotic cell. We have the gas vacuoles. Now, gas vacuoles are again those vacuoles which contain gas in them. So they store gas. They do not have an outer covering to them. Also, the gas vacuoles are present in the form of hexagonal gas cylinders. There are hexagonal cylindrical structures which come together to form gas vacuoles. So, hexagonal cylindrical structures which are called as gas vesicles. It is these gas vesicles which come together to form the gas vacuole. Going on to the next one, before that I would want to add that the gas vacuoles are permeable only to gases. They are impermeable to water. So they will not allow water to enter. So they are impermeable to water. And they are permeable to gases. Going on to the next one, which are the B1 are the inorganic inclusions. Or inorganic inclusion bodies. In inorganic inclusions, we have granules like those of sulfur, those of iron. So in inorganic, we have the sulfur granules, we have iron granules, and the third inclusion body are the organic inclusion bodies. Organic inclusion bodies will have granules such as those of starch, granules of proteins. So we have starch granules, we have protein granules, also we can add our glycogen granules. So these are basically the food reserves. Organic inclusions are the food reserves. Going on to the next structure in the prokaryotic cell, which are the plasmids. So number 6 will be plasmids. Now what are plasmids? Plasmids are basically extra chromosomal DNA. So there is an extra chromosomal circular naked DNA, which is again double stranded. So these are extra chromosomal double stranded naked circular DNA. Now these plasmids, they are capable of self-replicating. So they have this important property of self-replication. What are the functions of these plasmids? Now plasmids have certain important genes like the resistance gene, the gene which has the resistance factor, or gene which has the fertility factor. So it may have important genes. For example, the resistance factor, fertility factor. So this was about the plasmids. The next structure in the prokaryotic cell is number 7. Let us talk about the flagella. As I mentioned in the beginning, flagella are locomotory structures. They help the bacteria to be motile, to move about. So these flagella are actually tubular structures, or I can say tubular filamentous structures, which are responsible for locomotion. The structure of flagella can be divided into 3 parts, as we can see in the diagram. 3 parts are the basal body, there is a hook, and then there is a filament. And talking about the basal body, it is the basal body which forms the base of the flagella, and this is the part which is embedded in the wall. Okay, so basal body is in the wall and forms the base. It is forming the base. Then comes the hook. The hook connects the basal body to the filament, and it is a curved part. Hook is the thickest and the curved part, and it connects the basal body to the filament. Going next to the filament, as we can see, filament is again a tubular structure. It's a tube-like structure, and it is the beating of this filament which helps the bacteria to move around or to proceed in the movement. So that was about the flagella. Going on to the other appendages, number 8, pili and fimbriae. As I mentioned earlier, pili are the appendages which help in conjugation. So pili help the bacterial cell to attach to another cell and form a conjugation tube. So it is the pili which help to form a conjugation tube with the other cell, with the recipient cell. And again, pili is a small tubular structure. Talking about the fimbriae. Now fimbriae from one cell, there are many fimbriae on one bacterial cell or one prokaryotic cell. The fimbriae are basically small hair-like bristles structure. So they are hair-like bristles. And there are around 200 to 400 of them per cell. It is these fimbriae which help the bacterial cell to attach to the other cell. So it brings about attachment between two bacterial cells. Or two prokaryotic cells. So that was about the structure of a prokaryotic cell. Let us revise. We have the inclusion bodies, which are basically storage granules. They may be of 3 types. Gas vacuoles, which store gases. They are hexagonal cylindrical structures called as gas vesicles, which come together and form gas vacuole. These vacuoles are impermeable to water, but they are permeable to gases. Second type are the inorganic inclusion bodies, which could be the sulfur granules, iron granules, and so on. The third are the organic inclusion bodies in the form of food reserve. Now, organic inclusion bodies are mainly concerned with food reserve. So these can be protein granules, starch granules, or granules of glycogen. Coming to plasmids, plasmids are basically extra chromosomal DNA structures. So these are extra chromosomal double-stranded circular naked DNA. These are capable of self-replicating. They have important genes, for example, the gene having the resistance factor or gene having the fertility factor. Also, we have the flagella, which is a locomotory organ of the bacterial cell or the prokaryotic cell. These are long tubular structures or microtubular structures. With the body can be divided into 3 parts: the basal body, the hook, and the filament. Basal body forms the base and it is embedded in the cell wall, and the hook connects the basal body to the filament, and it is the thickest part of the flagella, whereas the filament is the long tubular part which helps to move about. Next appendages are the pili and the fimbriae. The pili are structures which help in building the conjugation tube between 2 cells, and it is a tubular structure again. Fimbriae are small hair-like bristles. These may be around 200 to 400 per cell. They help in attachment between the 2 bacterial cells. So that was about structure of a prokaryotic cell.